Good day, class. My name is Anna Gillett, and my group members and I, it is Celia Chun, Ari Fuller, Leanna Nicholson, Kylie Franklin, and Melvin Eunice will be presenting on DNA testing.
What is DNA testing? DNA testing is also known as genetic testing. Genetic testing is used to detect DNA sequence and the chromosome structure. Genetic testing can also include measuring the results of genetic changes, such as RNA analysis and an output of gene expression, or through biochemical analysis to quantify specific protein outputs.
The process of educating a person about tests and obtaining permission is called informed consent. Once a person decides to proceed with DNA testing, a medical, geneticist, specialist, or nurse practitioner can order the test. Genetic testing is often done as a part of a genetic consultation. Genetic tests are performed on a sample of blood, hair, skin, amniotic fluid, the fluid that surrounds a fetus during pregnancy, or other tissue. There are 4 main steps on how DNA testing works. First, it goes through the process extraction. DNA is located within the nucleus of cells throughout the body, and the extraction step is responsible for breaking open the nucleus and releasing DNA molecules into solution. During this step, it is also possible to separate the DNA molecules from all other cellular materials and any other debris that may be present in a particular biological sample. Some of these materials can be potential inhibitors to steps later on in the DNA testing procedure, so it is important to try and isolate only the DNA molecules. DNA quantitation is conducted to determine how much DNA is contained within a given biological sample. One of these standards all DNA testing laboratories must meet is to ensure that the DNA recovered from the extraction is human, rather than from other sources such as bacteria. This is done through quantitation, where the quality and the quantity of DNA present in a sample is measured and assessed. This process takes approximately 30 to 60 minutes to set up and then approximately 2 hours to run on the instrument. DNA amplification is accomplished through the use of a technique known as polymerase chain reaction, PCR. PCR is a process in which millions of copies of a specific sequence of DNA can be made in a matter of only a few hours. This process takes approximately 15 to 30 minutes to set up and then approximately 3 hours to run on the Thermo Scylla instrument. After the PCR reaction is completed, it results in a large mixture of amplified DNA molecules which need to be separated in order to distinguish the various molecules from one another. This is accomplished through a process known as capillary electrophoresis. DNA molecules carry a negative charge, and once an electric current is applied to the sample through the molecules enter a very tiny capillary filled with a gel-like polymer and migrate towards the positive anode on the other end of the capillary. The PCR products are then separated by size because the smaller the DNA molecules will have an easier time migrating through the polymer than the larger DNA molecules. The data from this process is then collected to a computer attached to the CE instrument, and then through the use of a software program, a DNA profile is developed. Setup for this step takes approximately 10 to 15 minutes, and each injection on the 3130 takes approximately 45 minutes.
I will be discussing the benefits of DNA testing. The first benefit is nutrition. Scientific data has shown that food may have different effects depending on the person. For example, metabolism of different substances may vary from person to person depending on their DNA. This is important when choosing a diet and lifestyle suitable for your body. Another benefit of DNA testing is health. Based on their DNA, individuals are able to understand what genetic mutations exist within it and are able to inform themselves on diseases before they develop. DNA testing, therefore, allows them to receive actionable information that will allow them to build a prevention plan against these diseases.
There are both pros and cons when it comes to DNA testing. Some pros include identifying health risks and diseases such as breast and ovarian cancer, discovering bloodline, which can help you learn more about your ethnic background, and it also helps in selective breeding in plants and animals. Some cons include ethical concerns such as individual freedom, health concerns such as the amniocentesis test, which can cause health risk in early pregnancy, and it also may result in inconclusive test results.
My name is Arful and I'll be discussing some examples of DNA testing. First of which is paternity testing, which is a definitive way of confirming a child's paternity. Second of which is in criminal investigations, where they will just simply take blood samples or fingerprints from repeated offenders in case they should commit another crime. And the last one would be prenatal genetic testing, which will just simply determine if an unborn child will have any health problems based on the genetics of their parents.
Hi again, I hope you enjoyed and were able to take some information from our video. This concludes our presentation. Peace.